that the repressor protein plays a very important role. Repressor protein, and I think that you are quite aware, and I think that it might be in your memory that it is the repressor protein which is encoded by the regulator genes. A regulator encodes a protein, and that protein is known as the repressor, and the repressor protein may be active or inactive. You know that in case of lac operin, the active repressor is produced, and in case of tryptophan operin, inactive repressor is produced. And I I I also hope that you are you know the term used for the inactive repressor, which inactive repressors are produced not only in the tryptophan operin, but it is also produced, it is also encoded in case of histidine operin. The regulator of histidine operin also encodes inactive repressor, and tryptophan operin also encodes inactive repressor. And the very important term is there for the inactive repressor, and it is known as the aporepressor. But in case of lac operin, the repressor is active, and active repressor directly bind with the operator and blocks the binding of RNA polymerase at the promoter region. You all know that that if the operator is not free, that is, if it remains blocked, or it remains bound with the repressor protein, the RNA polymerase cannot bind at the promoter. So for the successful or efficient binding of the RNA polymerase at the promoter region, there should be the operator should be free. So this is the first point of difference between the prokaryotic regulation of gene expression and eukaryotic regulation of gene expression is that in case of the prokaryotic regulation of gene expression requires the repressor protein encoded by the regulator, but in case of eukaryotic gene expression, there is no such protein. There is no such protein like the repressor protein which is used in case of regulation of eukaryotic gene expression. The very important difference between the prokaryotic and eukaryotic regulation of gene expression is that there is a requirement of repressor protein in case of the prokaryotic gene regulation. There is no requirement of any repressor protein like that of a prokaryotic repressor in case of eukaryotic. In case of eukaryotic regulation of gene expression, no repressor is required, no repressor protein is produced. Rather, on the other hand, the mechanism of the regulation of gene expression is different in case of eukaryotic regulation of gene expression. And you all know that in case of prokaryotic organisms or prokaryotic cell, the histone proteins are absent. Are absent, and there is no formation of any nucleosome model-like structure. There is complete absence of chromosome-like structure. Only the naked DNA is present, and all the prokaryotes are monogenomic and having a single genome consisting of double-stranded DNA only, and there is no formation of any nucleosome. So there are histone proteins are absent in prokaryotes. Okay? But in case of in case of eukaryotic organism, there is presence of histone protein. The eukaryotic DNA remains associated with the histone protein to form a compact structure, and that structure is known as nucleosome. So in this case, this histone protein also plays a very important role in the regulation of gene expression. You know that you also have the idea about the differences between the heterochromatin and euchromatin, which I have already taught you in your BSc Biotech Part 1, that the part of the DNA which is active is known as the euchromatin, the part of the DNA which is inactive is known as the heterochromatin. And heterochromatic region of the chromosome consists of compactly arranged or highly coiled and super coiled structure of DNA, and in this coiling, the histone protein plays a very important role. And for the transcription to take place, you all know that regulation of gene expression takes place by formation of the mRNA from the DNA. So if a particular part of DNA is to be transcribed, it should be free of the histone protein. It should not remain, it should be free. It should be accessible. It should be free to undergo transcription. So in case of prokaryote, there is no any histone protein, there is no association, there is no supercoil histone protein to check the regulation of gene expression and transcription. But on the other hand, in case of eukaryotic organism, there is presence of histone protein which remains in association with the DNA, and that histone protein is responsible for the compaction or the compact arrangement of the DNA to form a very important important structure which is known as the nucleosome. And the nucleosome, you know, that undergoes further coiling to form solenoid and super solenoid model. So if the DNA remains highly compacted, highly coiled, it cannot undergo any transcription, and regulation will, and the gene will not be expressed. So what will happen? What is the requirement for what should be for the what is the requirement for the transcription to take place in eukaryote if the DNA remains associated with the histone protein? The part of the DNA which is to be transcribed should get free from the histone protein. The histone protein should undergo disassociation from that part of the DNA. And the part of the DNA, or that is, you can say that the gene which is to be transcribed should get free from the histone protein. That the histone protein should remain, the histone protein should get detached from that part of the DNA. There should be disassociation of the histone protein from the part of the DNA which is to be transcribed. So what does it show? It shows that the histone protein also plays a very important role in the regulation of gene expression in eukaryote. So this provides a non-specific regulation of gene, because histone proteins are almost alike, either whether it is in protozoa, bacteria, protozoa, yeast, or the protozoans are the first animal, like amoeba and amoeba, trypanosoma, and all these things. So these are very simple eukaryotes, unicellular. But the histone protein found in the protozoa, amoeba, is similar to the histone protein and found in the human beings and higher plants. So this histone protein is the first. Histone protein plays a very important role in checking on suppressing the gene expression. How does it suppress it? If the histone protein remain associated with the DNA cannot undergo any transcription or regulation, cannot undergo expression by forming mRNA. So this histone protein is very important as far its role in the regulation of gene expression is concerned, because it provides, it suppresses, it prevents, the histone protein prevents the expression of prevent the expression of gene by preventing the transcription. Because if the histone protein remains associated with the DNA, that part of DNA cannot undergo any transcription. So what does it means? That histone protein plays a negative role. Histone protein suppresses or prevents the expression of eukaryotic gene. So what should be there? Whether the histone protein should be removed from that part? Yes, the part of the DNA which is to be transcribed at first will have to undergo disassociation from the histone protein. The histone protein should be removed from that part of the DNA which is to be transcribed. So this is very important, that the histone and non-histone protein which are associated with the eukaryotic chromosome play a very important role in the regulation of gene expression in the eukaryotes. And to explain the mechanism and the role of histone and non-histone protein and other proteins which are important in regulating the regulation of gene expression, three very important models are there: the Paul's model, the Frenster model which was proposed in 1965, and Paul's model, which was Paul's and co-workers proposed a different model in 1971 to explain the mechanism of regulation of gene expression. But later, the very important model uh, in this connection is the Breton-Davidson model. Breton and Davidson's were very important personalities who proposed a model to explain the mechanism of gene expression in eukaryote. And despite such so many models to explain the mechanism of regulation of gene expression in eukaryote, no model is still 100% correct. 100% correct or 100% um, correct in describing the mechanism. Still there is a lot of thing which remains to be unraveled and which remains to be understood. There is no complete idea about the regulation of gene expression in eukaryote till now. Still we have to develop, we need some very important models to be proposed to explain the mechanism of gene expression in eukaryote. But on the other hand, in case of prokaryote, the lac operon model, tryptophan operon, and all the mechanism of regulation of gene expression in prokaryote is well understood. And now, if it might have not been well understood, The Jacob and Monod would not have got the Nobel Prize. You know that Jacob and Monod were awarded Nobel Prize for proposing the lac operon to explain the mechanism of gene expression in prokaryote. So, as far our knowledge about the regulation of gene expression in prokaryote is complete with 100%. You have the mechanism of the regulation is fully understood in case of prokaryote, but in case of eukaryotes, the mechanism of regulation of gene expression is still not fully understood. All these models which I am talking about are although models, and they try to explain mechanism of regulation of gene expression up to some extent, but it does not completely explain the mechanism of regulation of gene expression. And so many models are there, but among these models, Breton Davidson